At 3 o'clock in the morning, your brain is shrinking, not metaphorically, physically. The cells that make up most of your brain, the neurons and the glia that surround them, pull themselves inward. The spaces between them open up by roughly 60%. And into those widened corridors, cerebrospinal fluid pours through, sweeping along the proteins your neurons dumped out during the day. One of those proteins is the same one that aggregates into plaques in Alzheimer's disease. Your brain does not rest at night. It runs its sewer system. The system that does this cleaning has a name that did not exist 12 years ago. In 2012, a team at the University of Rochester, led by Jeffrey Iliff and Iliff Nedergaard, pronounced Nedergaard, described a drainage pathway in the brain that nobody had properly mapped. They called it the glymphatic system, pronounced glymphatic, a word stitched together from glia, the support cells of the brain, and lymphatic, the fluid waste network that serves the rest of the body. 3 facts are enough to understand it. 1. The brain has no peripheral lymphatic vessels running through its tissue. It is the one organ that cannot be cleaned by the ordinary lymphatic system of the rest of the body. 2. Instead, cerebrospinal fluid, the same clear fluid that surrounds the brain, enters the tissue itself along the outside of arteries, exchanges with the fluid between cells, and exits along veins. 3. This whole process runs far more aggressively when you are unconscious than when you are awake. And everything that was taught about sleep before 2013 missed it. Here's what happens when you fall into slow-wave sleep. Step 1. The cells of your brain, neurons, and especially the astrocytes that cradle them, change shape. Measured directly in mice with 2-photon microscopy, the fraction of the brain occupied by the space between cells rises from about 14% while awake to about 23% during sleep. That is roughly a 60% expansion of corridors. Step 2. Cerebrospinal fluid is pushed into the brain along the outside of arteries. The driving force is not a pump. It is the pulsation of the arteries themselves. Every time an artery expands with a heartbeat, a bolus of fluid is shoved forward along its surface. Step 3. Water crosses from those periarterial channels into the interstitial space through specific water channels, called aquaporin 4 or AQP4, anchored on the feet of astrocytes. When those channels are missing, clearance drops by more than half. Step 4, the fluid, now mixed with whatever the neurons have shed, exits along veins and eventually drains out of the skull. This is a sewer. The brain evolved a sewer. You assumed, without noticing, that the brain cleans itself in the background, that there is some metabolic process handled by cells that quietly handles the garbage regardless of what you are doing. There isn't, not at the scale that matters. Measurements in mice show that the clearance of a radio-labeled protein called amyloid-beta, the one that forms plaques in Alzheimer's disease, is about twice as fast during sleep as during wakefulness. Popular summaries sometimes round that up to 10 times more active during sleep. That larger figure is a back-of-the-envelope calculation that combines the roughly 60% expansion of space with the increase in flow velocity. The literal number from the 2013 science paper is closer to a 2-fold acceleration of specific tracer clearance. But even that is an enormous disparity for a biological pump. It means the brain cannot meaningfully clean itself while you are running it. There is a reason for that. During the day, your neurons are firing, your astrocytes are swollen, and the corridors between cells are narrow. There is no room for bulk fluid flow. At night, the cells contract, the corridors open, and the arterial pulsation has somewhere to push the fluid. The system was designed, by whatever evolutionary pressure shapes brains, to run during the single state your body spends 8 hours in every night. When you skip that state, nothing substitutes for it. Not coffee, not meditation, not a walk outside. The sleep clearance connection is no longer a theory in mice alone. In 2018, a team at the National Institutes of Health, Ihsan Shokri Kojori, pronounced Shokri Kojori, and colleagues, scanned the brains of healthy adults after a single night of full sleep, and again after a single night of complete sleep deprivation. They used PET imaging, positron emission tomography, to measure amyloid-beta burden directly. After one night without sleep, amyloid-beta in the hippocampus and thalamus went up by about 5%. One night. This is not a small finding. The hippocampus is the region that loses function earliest in Alzheimer's disease, and 5% is the movement produced by depriving someone for just 24 hours in people who are otherwise healthy. A separate set of studies, led by Humberto Mestre, pronounced Mestre, at Rochester, has shown that the driving force of the whole system is the pulsation of the cerebral arteries, and that when arterial pulsation is weakened, with age or with hypertension, the glymphatic flow drops. A 2014 study by Kress and colleagues measured a 40% decline in clearance efficiency in older mice. The system works, it is measurable, and it degrades with age, blood pressure, and, most controllable of all, sleep loss. Sleep is not rest. Sleep is a shift change. Specifically, it is the window during which the brain swaps from one mode, running a conscious model of the world, firing trillions of times per minute, generating metabolic waste at full tilt, into a different mode. In that second mode, cells make themselves smaller, the corridors open, the plumbing runs, and the waste of one day is physically cleared before the next one starts. If you consistently cut that window short, the waste does not disappear. It accumulates slowly for most of a lifetime, and the evidence connecting chronic sleep loss to the proteins that drive neurodegenerative disease, amyloid-beta, tau, alpha-synuclein, pronounced synuclein, is now heavy enough that it is no longer a hypothesis in neurology textbooks. It is a risk factor. Your brain is not a machine that rests when you sleep. It is a machine that cannot clean itself unless you do. The exact physics of how fluid moves through the brain is still debated. Some groups, most publicly Alan Verkman and colleagues, have argued that plain diffusion alone is sufficient to explain the observed clearance rates, and that the glymphatic framework overstates the role of true bulk flow through the tissue. Others, Mestre, Nedergaard, Iliff, have replied with direct measurements of particles moving through the perivascular channels at predictable rates, driven by arterial pulsation. The phenomenon is real. The clearance changes with sleep. The amyloid-beta goes up when you do not sleep. The dispute is about mechanism, not existence, and the most important clinical question is still unanswered. Can we intervene? Can a drug, a posture, a breathing protocol keep this system running at 30-year-old efficiency when you are 70? At 3 o'clock in the morning, your brain is shrinking, and a fluid you have never felt is flowing through the gaps, carrying away proteins you will never see. It has been doing this every night of your life, and it cannot do it if you are awake. Every week, another piece of the machinery that runs your night. Subscribe. The next episode might change what you think you know about your own brain.